So our first speaker this morning is Dr. Thomas Pietschmann, who is the director of the Institute of Experimental Virology at TwinCore in Germany. Dr. Pietschmann studied biology at Julius Maximilian University Würzburg and Duke University in the States, and received his doctorate at the Institute of Virology in Würzburg on the molecular biology studies on the function of the envelope protein of human foamy-virus. He then moved to the University of Mainz, where he began his work on the replication mechanisms of hepatitis C virus, and in 2006 set up a junior research group at Rupert Karls University Heidelberg as part of the Emmy Noether program. A year later, in 2007, he accepted the call to Hanover and has been researching there ever since at TwinCore as the head for the Institute of Experimental Virology. Dr. Pietschmann is an elected member of the Scientific Advisory Board of the Society for Virology and of DFG Panel 204, Microbiology, Virology, and Immunology. He is a speaker of the DZIF Hanover Brunswick site, and since this year is the scientific co-director of the Helmholtz Center for Infection Research. So without further ado, Dr. Pietschmann.
So, bonjour, and good morning. Very honestly, I've never been at a meeting that has been so respectfully, friendly, opened like this one. So it's a pleasure to be here, and I appreciate this very much. And it's really special. And so I hope I can contribute to this meeting a little bit meaningful information about hepatitis C virus, vaccine research and development, and some of the work that we do. And I very much thank you for the invitation to be here, all the organizers. I don't name anybody specifically because there's so many involved, and thank you for having me. So what are the challenges in HCV vaccine development? I would also like to talk a little bit about the opportunities of this, not only the challenges, and what are the limitations of our models that we use to drive forward vaccine research. Before I go there, I'd like to show you a few pictures I took yesterday when I flew into Quebec. I was really impressed to see some of the aspects of the city, including the parliament I learned. Then, of course, the beautiful river, St. Lawrence Stream, we call it in German, with lots of ice on it, quite amazing. Then this castle-like hotel I found out. And then I learned about the tragic story about these two soldiers who gave their life in a fire in Quebec, saving other people's life. And I also saw these women in front of the parliament, but I don't know their story yet. So maybe there is time that somebody helps me find about these women here. So with this introduction, I'd like to directly jump into it by using this bold statement of our Nobel Prize laureates from 2023 for the RNA vaccine development. Kathleen Kareiko and Drew Weissman says, we're now working on all sorts of vaccines in everything using the mRNA platform, and I think it's a breakthrough, a major breakthrough that was honored, justified. But we'll see how easy it is going to be to develop vaccines against all these pathogens. And I'd like to move on by sharing this slide with you, summarizing a recent publication of the WHO, guiding us where is really particular need in endemic pathogens, not the emerging ones like SARS, but those that have been with us for a long time. And this lists, among a few others, also hepatitis C virus, based on a matrix of characters such as socioeconomic burden, severity of the disease, disease burden, epidemiology, and so on. So I think we are on the right track to work and join forces towards development and implementation of a hepatitis C vaccine. This now brings me to the challenges, and I'd like to start maybe with the most daunting one in the beginning, and I'd like to introduce you to Stuart Ray from the United States, who gave me this slide showing the vast diversity of hepatitis C virus strains that circulate on this planet. This genetic tree shows by distance how distantly related different strains are, and you can see that the hepatitis C virus family tree is much larger than hepatitis A or B or even HIV. And if you amplify the SARS-CoV-2 by 70-fold, then you can see it on the map. If you were not to amplify it, you would only see this little spot. And you know, in the SARS vaccine development, we have to reprogram the vaccines every year to match the new variants. So that shows you the challenge of getting a pan-protective hepatitis C virus vaccine, given the wide diversity of this pathogen. Challenge number 2 and 3 I want to point out are listed here. The challenge 2 is illustrated here in a new structural biology work from Denmark that shows the structure of the envelope protein E1 and E2. It's the spike proteins of HCV. And if you look closely and compare where this, what is it in English, this color that I'm pointing out, you know what I mean, the brownish color. This is depicting a very good antibody that tries to block the virus, but you can see that this antibody coincides with portion of the envelope structure depicted in lila here. So that means that in a normal condition, this antibody would not be able to bind. Something with the envelope has to move away. The epitope is actually hidden, and that only occurs after the first entry factor has been met on the cell surface. But then the virus is already bound to get in, and it's difficult. You need lots of antibodies to then block it. But this clash here tells you that it's difficult to raise antibodies because the epitope is not there. It's not visible. So these hidden epitopes, the vulnerable spots of the virus, are hidden, makes it difficult to induce massive antibody responses against this virus. The third challenge is depicted here, and it's the nature and maybe the beauty also of the virus, but it's the peculiar nature of the virus to traffic in the bloodstream as a lipovirus particle. So it's not only composed of viral components, but also of human lipoproteins, so our own proteins that help the virus to disguise and not be visible to the immune system on one hand, but also that helps the virus to directly recruit to the liver because lipoprotein receptors are resident on hepatocytes and help the virus to get in. So that brings me to even further history in the story of looking at HCV, discovering it. That was done by Michael Houghton, now in Canada. He got the Nobel Prize for his discovery in 1989. And then it took more than 20 years to learn more about the virus structure with Charlie Rice, who is also the co-discoverer of Hep C, also got the Nobel Prize. And he published these peculiar pictures of hepatitis C viruses, where it looks like a ball in a ball, like maybe a virion in a larger lipoprotein ball, as we believe. And that's the best pictures that we have. And that took 24 years to discover, lots of work. And if you imagine then, each of you knows the Zika virus, where we also had a pandemic, an epidemic. The WHO called for the emergency in 2016, and then in April 22nd, 2016, already the first structure of Zika virus was published. So it's actually like 2 months only. And Zika virus is related to hepatitis C. It's also flavivirus. It has, different from HCV, this golf ball-like structure, highly regular. The envelope proteins are all clearly visible on the surface of this in red and yellow. And so it's much easier to derive this structure, likely also a vaccine against this virus, because it doesn't have these peculiar adaptations that hepatitis C virus evolved and that help HCV to cause a chronic infection, because these adaptations help it to evade and even to circumvent a powerful immune response to be established. I'll come back to the work of Charlie Rice. The pictures you see again on the left, where it's suggested that a particle is within a particle, and when they purified these virions out of a large production of hepatitis C virus particles and analyzed the viral proteins in the virions, they found, of course, as expected, the viral proteins, E2, the envelope protein, the core, the capsid. But what they also found was, I think I have to return this way, these lipoproteins called apolipoprotein E and apolipoprotein A, they are integral components of the virion. They're directly in the virus particle, and they shape its features, how it enters cells. Something else I want to mention is that HCV, and that's also distinguishing it from other viruses, changes its composition and function in the bloodstream of humans, of animals, and even in cell culture, if you wish. And that's depicted here in this work from Brett Lindenbach, from Charlie Rice's group, who looks at the density, that means, the composition of HCV particles in tissue culture, when they were then used to infect chimpanzees, and then when they came back to tissue culture. And you see the buoyant density of the virions here, and then this is where the viral RNA is, and you can see a peak here, and that's roughly at a density of 1.18. If you go from this dot over here, you can see that. In the chimpanzee, in the serum, the virions have a much lower density because they interact more effectively with low-density lipoproteins, so that the majority of viruses have a lower density at around 110. And then the virus comes back into cell culture. If you collect it from the serum, infect tissue culture cells, then the virus goes again back to higher density, showing how it shifts depending on the environment. And that has to do with exchangeable apolipoproteins, such as ApoE, moving and getting off from the virion, depending on the abundance of these lipoproteins. This is not only occurring in animal models or in tissue culture. The group of Thomas Baumert has shown that also this occurs in humans and human HCV patients. So that was a study published some years ago where they gave a high-fat diet meal to HCV patients and then tracked the virus density over time after the meal was ingested. And you can see that over time, as about 1 or 2 hours to 3 hours after ingestion of the meal, most of the virus is at a very low density fraction because it interacts more with very low density lipoproteins, and then after the meal has been digested, the virus goes back to a state where not so many viruses have a low density. So this shows you the virus particles change over time. And I said this is functional relevance, and I'd like to show you an experiment from in vitro, also from Thomas Baumert's group, showing that that has really relevance. What we can do in tissue culture is we can manipulate the system. We can transfect silencing RNAs into the virus-producing cells and disrupt the expression of apolipoprotein E. So less apolipoprotein E is available. What happens is depicted here. If you then take the viruses from cells that have low apolipoproteins, liver cells that can't produce so much of the lipoproteins, you can see that these viruses are more vulnerable to neutralization, about tenfold. You can neutralize them with human antibody-containing sera against HCV about tenfold more. That means the virus is losing some of its protection if you take away the apolipoprotein E. On the opposite, and that is some work that we have done, is you can also add apolipoprotein E to the virions and then see what happens. And what happens is depicted here in these neutralization assays in tissue culture. The light blue line is when we incubate the virus with additive apolipoprotein E in numbers similar as would be present in human sera. You can see that the infection goes higher, the infectivity of the virus is enlarged, and it's more difficult for these powerful antibodies to diminish the infectiousness of HCV. So that's seen in these curves, showing that if you have more lipoproteins loaded onto the virion, then it's very difficult for antibodies to neutralize it. And this little picture here summarizes this phenomenon that HCV replicates in liver cells. It is secreted with lipoproteins. Additional lipoproteins that circulate in the blood add onto the virion to make it a mature virion heavily decorated with lipoproteins. And these facilitate the attachment to liver cells and thereby the escape from antibodies. So this is what we currently know, and so we need to use tissue culture models taking this into account and use them as a proxy to measure antibody responses to the virus and hopefully also vaccine-induced antibodies that would hopefully be powerful enough to protect from an HCV infection. The remainder of the talk, I will talk more about the opportunities in HCV vaccine development, and the first one is what the virus does with us itself, because most of us, not most of us, but 1/3 of us, if we were exposed, can clear the virus naturally. That means we have endogenous immune responses powerful enough to prevent the infection. And the disease only occurs after decades, so that would be sufficient. On the opposite, it means if you learn exactly what helps us to be protected in the first couple of weeks, we would have a blueprint for what a vaccine would have to do. So it's worthy to investigate that further. What are the protective mechanisms that lead to clearance? Others, and also we, have done this in the past couple of years, and it's clear that antibodies play an important role. Antibodies that combine very different variants of HCV, because as I said, HCV is highly variable. But there's strong evidence from several groups that early induction of such antibodies correlates with clearance in the acute phase. And then it's been shown that these antibodies are the most powerful that bind to an envelope protein portion that binds directly to the receptor CD81. It's very similar like in SARS, where the ACE2 receptor and those antibodies that prevent seeing the ACE2 receptor bind and that prevent infection. It's also been shown in animal models that if you only add antibodies to the animals, they would not be infected. And it's also interesting that there's not so much mutation in antibody genes that is necessary to get very powerful antibodies. I apologize for focusing this slide only on antibodies. I have to add also T cells are very important, of course. Broad and broad-specific T cells also are essential to induce antibodies and to directly kill infected cells. I specifically focus on antibodies because the assays I explain now measure antibodies. That's why I wanted to introduce the role of antibodies, not neglecting the other important functions of the immune system that are also important for vaccine development. The third opportunity, again related to antibodies, is depicted here, and that is that we begin to understand the structure of the target, the envelope E1E2 protein, the spike of HCV. And if you know how it's shaped and what the structure it has, then you can find out where do the antibodies bind, how do I have to prepare an optimized antigen that presents these vulnerable epitopes. This is a side view of the E1E2 heterodimeric complex. It's like in a crisscross like this. On the right, you can see it from the top, and here you can see it embedded in the viral membrane. I'd like to share this area in orange. That's the CD81 binding loop. This is a key feature of the envelope where it binds the receptor, so antibodies against this area are critical. And we need to have antigens that expose this in a very stable way to have many antibodies. So that brings me to some of our own work that I wanted to share with you in the end, with beginning the strategy that we use. So first, in our lab, we want to develop good assays that measure immune responses to HCV, both in vitro, but also in vivo. I will not talk about our work on developing an animal model, but that's also something that we'd like to do so that you could test in an animal model, do I have a vaccine that really protects from an infection? I focus on the in vitro models to test immune responses in humans. And then towards the end, I will talk about different vaccine platforms that we use to induce powerful antibody responses. This is a bioinformatic virology work that I try to summarize in a few sentences. What we did here is we took 101 polyclonal antibodies from human patients infected with diverse viral genotypes to see the diversity of antibodies that humans mount against HCV. And then we took 13 different viruses of HCV representing all the genetic diversity or much of the genetic diversity of HCV. And we tested how well would these antibodies inhibit each of these 13 viruses. So you have data from 13 viruses against 101 antibodies. And then the bioinformaticians plotted only the viruses in a grid that you can see here, showing how related the responses of the viruses to these 101 antibodies were. And viruses that are highlighted by the small dots that are close to each other had a very similar reaction with these antibodies, were similarly neutralized by this and that, but not by other antibody preparations. And viruses that lie out here are very different. They are neutralized by another subset of antibodies and not the others. So you get this landscape of how viruses fall into different clusters. And that helped us to say, we don't have to always screen with all these different genetically diverse viruses. We take a subset representing the 6 distinct clusters, showing the distinct biologies of HCV viruses as they circulate in humans. So we then moved on only with these 6 reference viruses, which makes it a much faster and a relatively balanced assay to look at how HCV responds and is inhibited by antibodies. So that helps us to look at how antibodies neutralize HCV, and it helps us also to benchmark vaccine-induced antibodies, how powerful they would be relative to antibodies that humans do, and how that hopefully delivers a protection. In collaboration with other teams, we've now used this panel to identify some very good neutralizing antibodies that matured in human beings, and the structural biologists in the team could show how they bind the E1E2 protein a little bit in a different way than other antibodies that have been described, and clearly also where the CD81 receptor binds the HCV E2 protein. And they have a specific type. So it's not such that all of our B cell repertoire responds to the virus, but there's unique heavy chains, we call them, that are responsive, and one of them is the VH169. So it's important then to look whether our vaccines would induce such type of antibodies that would likely make them relatively protective, because these are the antibodies used by the most powerful naturally induced antibodies. Then coming to the vaccine efforts that we have been moving forward in the last couple of years, we first said HCV is so diverse, and maybe some of the antigens that HCV has in the one or the other strain are more suited for a vaccine than others for biological features that open the epitopes more openly to antibodies and that make it more immunogenic. And so we selected in total 12 different HCV strain antigens, because some of them have different contacts to entry factors, as depicted here on the left. Some of the viruses can only use one of the receptors, others can use different claudin receptors, as indicated here. And some of these envelope proteins are either very resistant to neutralizing antibodies, or very vulnerable. So they have different biological features, and we just tested exploratively whether they would be more, the one would be more useful than the other. And then we also said it's not really known which vaccine platform, and there's many these days, viral vectors, protein-based vaccines, mRNA-based vaccines, would be most suitable to induce antibodies against HCV. So we tested some of the variants in all these different platforms to then know what is the best combination. And since this is an in vitro assay, we said we have to somehow benchmark it with human data. And so we used all the patient sera that we have screened over the years to get an imprint, what is the most powerful antibodies that humans over decades of an infection could mount, and how do the vaccine-induced antibodies match to this potency. The heat map shows of a couple of hundred patients that we tested against the reference viruses, those patients at the top that mount the best antibody responses under these assay setups that we use in vitro against all these 6 reference viruses. That's the colorful dots, and the mean is here. So the best human beings that we tested neutralize 80% of the infectiousness of all these 6 reference viruses under these assay conditions. So that's quite powerful. And some of the patients mount not very good antibodies at all, although they have been infected for many, many years. And then we also had access, thanks to Michael Houghton, who gave his clinical state vaccine candidate and the sera that were collected from a human study, so that we could test the vaccine-induced antibodies from the most advanced humoral vaccine that has been tested in humans. And there you can see the placebo. As expected, none of the participants mounted any neutralizing antibodies to any of these 6 viruses in the placebo group. But then you see a low but measurable antiviral effect of the antibodies induced by this vaccine against the different 6 test viruses. The yellow one here, the most powerful at 20 microgram dose. And if you bulk this, all the participants, into a mean value against the reference viruses, you can see a significant dose dependent reduction, although it's moderate. And that is the blue line here, so that's the Houghton vaccine in phase 2 trials in humans. And the red line is the most powerful antibody responses that are matched in humans after a decade infection. And this is our vaccine screening of diverse envelope proteins of HCV. And you can see that there's not much difference. So HCV has simply evolved not to have very immunogenic antibody-inducing E2 proteins, but H77 is the one that Michael Houghton used. This is the one that we use. It's similar range. It's not much different. If you combine the best E2 proteins in one cocktail, it goes down a little bit further, but it's still a moderate neutralizing antibody response. And of all the screenings, our most powerful candidate that we now follow up is a VSV vectored E1E2 protein. So that's a viral vector vaccine. This is licensed for Ebola vaccine, so that's a licensed vaccine, and we now moved on the envelope proteins of HCV E1E2. And that's a mouse experiment where you see all the mice responded to the vaccine candidate to a quite substantial level. And if you average across the reference viruses, you can see that delivers an immunogenicity of antibodies in the range of the 5% best human antibody responses measured in the same assay. So we find this is encouraging, and that is what we now follow up for onward development in our vaccine program. With this, I'm now at the end of my talk, and I'd like to briefly summarize the main points. I started out with the challenges, again listed at the left side: genetic diversity of HCV, the lipoproteins that facilitate entry and help evade antibodies, and the hidden epitopes where antibodies have a clash and can't really bind. The opportunities, on the other hand, that many of us would be able to autonomously clear the infection. Protective immunity is possible. The key role of antibodies in protection, next to also T cells, of course, and the structure of E1E2 proteins that is emergently helping us now to be on target in our vaccine development. We have developed a reference virus panel that helps us to measure the breadth and potency of antibodies, and we use it as a benchmark to rank order our vaccine candidates for onward development. From testing 5 different vaccine platforms with diverse envelope proteins, we conclude that the VSV E1E2 is the most powerful candidate so far, and we'd like to bring this for onward clinical development forward now. So I'd like to acknowledge many people. I would like to start in my team with Dorothea Bankwitz, who used to be a PhD and postdoc in my lab. She's now the project manager in the vaccine development work. The key structural biologist that helps us making vaccine design is Thomas Krey from Lübeck. We work together with clinician scientists Markus Kornberg and Anke Kraft from Hannover Medical School, who helped us and get access to the patient sera. Florian Klein from Cologne is an expert in monoclonal antibodies, helping us to clone all the monoclonal antibodies from humans. We are grateful for the collaboration with Michael Houghton, giving us access to the human sera with the previous vaccine trial. And Daniel Pincheva, Gerd Zimmer, and Aziza Folz helped us with the different vaccine platforms. And the bioinformaticians are Alice McHardy and Chris Lauber with their teams. We're grateful for the funding. And also, we can now give out the reference panel thanks to agreements with Torre and APATH. I'd like to end with a forward-looking statement here. My institute is associated with the Helmholtz Center for Infection Research in Germany. That's a larger structure, and we are inviting people who are curious in infectious disease research. We'd like to cite the work of Françoise Barré-Sinoussi, who coined this very nice phrase, I think: the fight against infectious disease is never ending, and each generation has its role to play. How true. We'd like to use it and also pinpoint the role model Rosalind Franklin by inviting young people for PhD applications if they want and are interested, that we have just published, to be curious in infectious diseases and help us tackle them. Thank you for your attention.
Thank you, Thomas. That was a really nice talk. So I think we have time for a question or two. I think there's one in the back corner.
Yeah.
I wonder if we're not shooting ourselves in the foot by showing this diversity slide. And, you know, I love Stuart. Don't get me wrong. I've worked with him for 25 years. But I wonder if we're not shooting ourselves in the foot, because those are not serotypes. And in fact, our studies of people who are at ongoing risk of HCV infection show that those who spontaneously clear, as you mentioned, have really robust evidence of protective immunity. And I'll point out that that's pangenotypic immunity. I can't say it's totally pangenotypic, but they've cleared 3 different genotypes and multiple subtypes. So I think we may be selling the diversity a little bit harder than is necessary. I think the major barrier to our having a Hep C vaccine is the complete absence, with one exception, of any funded efficacy trials. And so we've taken a different strategy, which is we see people who are infected have some commonalities of the infecting sequence when they clear. And we've now partnered with Moderna to develop vaccines based on those sequences. So I just wonder about your thoughts about putting this diversity out there first, because I think it sets people into this negative mindset when in fact there is really cross-genotypic diversity.
Thank you for this comment. It helps me also to clarify this further, and I agree with you. the variability of HCV is not this great across all domains of HCV, and there are some highly conserved areas where the virus cannot move. This would be ideal for vaccine development, and likely immunity to these highly conserved regions will deliver protection. Also adding to this, the natural clearance in the acute phase of 1/3 of the patient indicates that it is possible to mount protective immunity. So I agree with you, and that has now nicely been adjusted.
And the second comment I wanted to make, thank you for developing your CC panel. Justin Bailey worked with people all around the world to develop a pseudoparticle panel because it would be perceived as biased if we evaluated other people's vaccines when we have our own candidates. That's been transferred to Duke University, and so you can send serum to them and they will test for breadth of neutralizing antibody using this reagent which we've supplied to Duke.
Absolutely. This is a parallel system that's equally suited.
Okay,
So
I think in the interest of time, we actually have to move on. Sorry.
Thank you. Yeah.
But thank you, Thomas.
Thank you.